symposium, second day. I believe yesterday, as many of us that were able to join, we learned a lot of things, and we went home with things that will make us better researchers in the field of bioinformatics. So today is our day 2, and we are going to start with an opening prayer to be led by Dr. Moses Ikebunam. Dr. Moses, please lead us in opening prayer.
So we pray, Father, that you be with us today in all our presentation, in all our deliberations in the field of bioinformatics, grant clarity to all the presenters, and grant us understanding. Pray through Jesus Christ. Amen.
Amen. Thank you so much. So the next thing we'll have is national anthem.
Okay.
Thank you so much. And now we take the Nnamdi Azikiwe anthem.
Chromebooks haben bis zu 12 Stunden eine Akkulaufzeit. Also welche
Film schauen wir als nächstes? Wechsel zu Chromebook.
Pride of the states and our nation, fountain of
Thank you for that anthem of the University of the Moment. And now I hand over to the technical section of the person that will pilot us through the technical section, Dr. Akinola Adekoye. Dr. Akinola, please unmute yourself and take over. Dr. Akinola.
Okay. All right. Good morning, everyone. Everyone hear me?
Yeah, we can hear you.
All right. Okay. Good morning, everyone. Thank you. Thank you for that brief introduction, and then for the event for today. We thanks for yesterday and everyone that showed up. And this morning, we'll be moving straight to the technical session, which I'll be the chair. And I would like to just let the presenter know that we'll be strictly adhering to time so that we'll be able to finish at the allotted time. So this morning, without further ado, I will be introducing our first speaker for the day, which is Dr. Samuel Ayodele Egiye. Dr. Samuel is actually a seasoned and highly experienced pharmacist with bachelor and master degree in pharmacy and a PhD in bioinformatics. He also has a postgraduate diploma in clinical research and drug development from the University of Basel in Switzerland. And then he's currently a senior lecturer at the discipline of pharmacology and clinical pharmacy, School of Pharmacy, University of Western Cape, Cape Town, South Africa. He leads the computational and pharmacology and chemoinformatics research group. His research focuses on computational drug discovery and design, data science, which includes chemoinformatics, bioinformatics, and machine learning for predictive drug development, for infectious disease and precision medicine. So, Dr. Samuel, you can take it from there.
Yes, sir.
It's over to you.
Okay.
You have the floor.
Can you hear me?
Yeah, we can hear you loud and clear.
Oh, beautiful. Thank you so much. Apologies, my video is not working today for some reason. I want to thank the organizers for giving me the opportunity to speak. Just to clarify, do I have 30 minutes or do I have 20 minutes?
It's 20 minutes.
It's 20. Okay, thank you. So I can run.
Yeah.
All right. Okay, I'm trying to change my slide here. All right. So briefly, I'm going to talk about my career path, and my focus is on drug discovery using those techniques that have been outlined in my bio. And I'll also look at the status of drug discovery, the pipeline, challenges in drug discovery, and asking the right questions in that space, and also tools that are already available that one can use to answer those questions. And I'll also go through a few case studies as much as I can at the end. My goal here is that at the end of this talk, for as many that are new in the field, should be able to run at least hypothesis generating or data analytic research using just their laptop and the internet in drug discovery. And I think that's the best place to start, rather than just waiting for everything to be perfect in the situation that we find ourselves, specifically in the country that we are in, and even in Africa. Okay, so in terms of career path, this is where I started out after leaving school a couple of years ago at the National Institute of Pharmaceutical Research and Development in Abuja. I was a junior research fellow there for a while. Then I during the HIV/AIDS era, I joined FHI under a particular global HIV program. I was there when I got a fellowship to come to South Africa, where I joined the International Center for Genetic Engineering and Biotechnology. While I was there, I had a stint with UWC doing some things, some contract work for them and teaching in the School of Pharmacy. Then eventually I joined SAMBI, where I did my PhD. While I was in SAMBI, I had an opportunity to visit Novartis under the Next Generation Scientist Internship Program. And that was an eye-opener. From there, I came back, wrap up my PhD. Then I had another opportunity to visit EBI in Cambridge. And that's also changed the trajectory of my career. All right, so let's get into what I'm going to talk about today. So what is the status of drug discovery and development? And I think this not only applies to Africa, but generally to all countries. The current paradigm is not addressing the treatment priority for developing countries. And we know that between 1975 and 2004, less than 2% of the new entities that were discovered were for neglected disease and tuberculosis. Of course, the big pharma, of course, are looking for more lucrative markets. And if you look at it now, genetic diseases are the more lucrative market. The population that are involved might be small, but they are high-paying populations. So rather than some other diseases that the population infected are more, but they are low-paying populations. Okay, so there's need to develop capacity in especially endemic areas that are endemic with this neglected disease. So we need to find a way to explore what we have in order to do that. Now, the pipeline of drug discovery, I'm sure you've seen this, and the emphasis here is to point to the fact that where we are in terms of basic research, and I'm trying to see if I can get a pointer, in terms of basic research, and we know that eventually what makes it to the market, it's quite very small compared to what you started with. So there's a high attrition rate in the pipeline. We're familiar with that for those that have done drug discovery. And the failure which leads to this attrition rate might be due to the toxicity of the compounds that were discovered or lack of efficacy, probably due to the fact that the target was not validated, or in issues around reproducibility. We're just making the same class of compounds, nothing new. So somehow we need to find a way to de-risk this process. And it's costing so much to run this process. And I feel that data analytics and computational modeling has come into play to find a way to reduce these attrition rates and somehow reduce the cost. These are the questions I want to attempt to answer in terms of current challenges when it comes to drug discovery. And in the recent paper, they highlighted a couple of these challenges. The first one being insufficient knowledge of underlying pathophysiology of diseases. Yesterday, when Dr. Ginola presented, he showed a very nice pathophysiology or the pathway for the work that he did. So we need to understand the pathway. And secondly, the lack of valid biomarkers or surrogate endpoints. How do you know your drug works if you don't have a biomarker that actually tells you something has happened in the system? Sometimes non-translational from the animal models. Of course, you need to start your basic research in animal models, and sometimes those results are not translatable to human models. Even when you translate to human models, the human population themselves have heterogeneity in their system. So some people will be it will be effective in some people, it will not be effective in some other people. So we need to be aware of that. Target identification is also challenging, really finding, and that is linked to understanding the pathophysiology. Then exploring what we call a biologically relevant chemical space. The chemical space, I'll talk more about that. Inadequate academic industry collaboration. That's one thing that's, the academics seems to be flying solo, especially in Africa. Then I will address the issue of training. So asking the right question, I think, is the beginning of research. Isaac Newton didn't ask, why is the apple red or why is the leaf green? But the question he asked that changed the world was, why did it fall down? And I think that is key to the things that we do. Now, once you have the right questions, there should be tools or avenues for you to answer those questions. And I think that's why we are gathered here today, looking at those tools and those fields, and bioinformatics, chemoinformatics, and data analytics, which are my core areas, are tools that I feel that can help us answer some of those questions in drug discovery. So in the next couple of slides, I'm going to ask some questions and try to highlight tools, databases, and things that I feel are available out there that we can use to answer those questions. So if you ask yourself, what is the underlying pathophysiology of the particular disease that you are interested in? You really want to know, understand it. And like we said, this is one of the challenges that we have in drug discovery. There are quite a number of tools out there to use, especially these tools that associate your gene with the disease. So what I've done on this slide is to give a couple of those examples. So for example, the OMIM is a comprehensive authoritative compendium of human genes and genetics phenotypes. And you can actually search this using any disease or gene or whatever clinical features that you know, and it will output genes that have been linked to that particular disease or clinical feature. Now, if you take output from there and run it through KEGG, which is a pathway database, it can also give you where those genes are within the biochemical pathway. This is another very good tool, a database that is well created, that also combines links to genes to associated human diseases. So when I did a test on that, I tried to explore hypertension using that platform. It comes out with, this is just one of the maps that it comes out with. And it gives you all the genes that are linked to that particular condition. And I picked one of them and I ran through KEGG, and I could see the pathway where that particular gene or the enzyme or the protein is involved with. So already you have an understanding of what the thing is, and you want to begin to think about, you can imagine the kind of data that you're going to generate from this for each of these genes that have been shown to be linked to that particular disease. So you have a lot of targets to work with in this particular scenario. The other platforms that have documented output from genome-wide association studies, the professors mentioned this yesterday. And when I tried this GWAS catalog and I ran the same disease, hypertension, I could see an output of studies that have correlated some genes with that particular disease. And you could see not only the genes, but the variants that have been linked to that particular disease, people that have a specific variant, and you can begin to explore those variants and see quite interesting things coming out of that. Now that you understand the association between the genes and the disease, the question now is, what are the druggable targets for that particular disease? What am I actually going to modulate? What do I want to modulate with my compound in order to produce a particular effect? So a lot of study has been done in gene knockout, gene knocking strategies, and a lot of them have been documented. For those that are working on infectious diseases, TDL target is a very good platform to use to begin to explore your pathogen and trying to look for druggable targets that have been curated in that particular database. And it allows you to select your organism and allows you to auto-filter those targets based on some features, some properties that really kind of gives reliability to the targets. Therapeutic target database is a collection of human targets that have been linked to diseases. Okay, so once again, these are tools that are databases and tools that are out there that have curated works that have been done in the field of bioinformatics. So now that we have the targets, what endpoints do we want to measure? What are the endpoints that you want to be able to see? MarkerDB is one very good database that allows you to, for a particular condition, allows you to look at what are the endpoints, what are the biomarkers that have been clinically or experimentally linked to that particular disease condition. In this case, once again, I tried hypertension, which is a common disease in Africa, and it gave me a couple of, of course, we know you need to take your blood pressure and the rest of them, but there are other biomarkers that you can also use to monitor or evaluate that particular disease. And with this database, it's just one that I've curated those biomarkers that are linked to a particular clinical feature or manifestation. Then the issue of models, animal models. Of course, we know we use quite a number of rats in Africa. Oyster rats, a lot of publications come out with oyster rats, and sometimes we grow organisms in petri dishes. So we try to do all this bioactivity assays and try to correlate them to what we thought would happen in human. And of course, there's no direct correlation, especially when we do toxicity studies sometimes. A lot of things are happening in this space, trying to use humanized mouse, zebrafish, and the rest of them. Those are improvements. But there are platforms that are now being designed. I just came across this recently, and I was engaging these guys in some conversation. So what they've done here was to curate or to take all the data that have been, all this clinical data or experimental data from animal studies, run them through a computational model and correlates them to a particular phenotype. So what you want to do is to be able to say, you have not done the study in human, but can I infer based on this data that is available what the phenotype or the predicted human effect will be? So they've come up with this machine learning model and that is able to take that data, and if we are able to, it's not free at this point, able to get your data in there, you can actually predict what your data will look like in human using their platform. Okay, so that's one way to translate animal models to human features. Another thing is, after you've done that translation, even if you do succeed in doing the translation, is the fact that humans are inherently different. We just find that in the field of pharmacogenomics and precision medicine, So FARM-GKB is another database that I've curated known SNPs and variations in human genes that are associated with particular clinical feature or particular, what's the word I'm looking for, response to a particular drug use. For example, so what I've done here was to look at a particular variation or that have been known in a particular gene that can lead to increased response to diuretics or hydrochlorothiazide that is used for hypertension. Okay, and this database is quite very, very useful if you begin to think how your drug will look like even before you start your clinical trials in humans. Okay, another guy that I recently came across that I've been talking to is this guy, SmartTR, SmartR. What they've done also is to look at what has been done in the human and running it through some machine learning platform. They've been able to isolate and predict the specific population that will respond to a particular drug. It's a bit more, this is an oversimplified picture, but I just could draw your attention to check them out. I think they have a paper out. You can also see what they did in that paper. But this is also possible in order to identify specific populations that will respond to your treatment. Okay, now, having said all that and you've been able to answer those questions, how do you now explore the chemical space? And I said biologically relevant chemical space because chemical space is very, very large. But you want to find those compounds, those stars, let's call them stars, or those things that will relevant to a particular disease. How do you navigate that space? Because you can't afford to synthesize the whole chemical space to explore. Now, people have done quite a lot of work in curating compound or ligand databases. Zinc is very popular. That contains large amount of purchasable compounds that people have synthesized over the years. You can buy them, you don't need to synthesize, and that's a very good way of saving some money. And AfroDB is another. AfroDB is now being being cataloged in zinc. It's a collection of natural products. A couple of guys have come together to do that for Africa. And also you have databases for protein. So ultimately, in drug discovery, we are always after a compound modulating a particular enzyme that is involved in a pathophysiology for a particular disease. That's what we want to do. So you have databases of compounds, you have databases of possible protein. UniProt has a very good curation of proteins and their link. That's very nice Go terms that tells you how they're linked to a particular biological process. Now, if you have done what we call phenotypic screening, maybe you just took an extract or isolated a compound and you ran it through a particular assay and you got a readout, you don't really know what your target, what your compound has eats in the system. But using some modeling prediction tools, you can actually have an idea of possible targets that your compound has eat. So using these kind of platforms, Swiss target prediction is a very good that has been used over the years, but quite a number of other platforms are also out there that allows you to predict possible target for your compound. And that gives you an idea to say, okay, can I modify this compound and see if I have improved activity, if this is the predicted target for that compound. Another thing that you can use to screen, instead of before going to the lab, at least to generate some sort of hypothesis, is your virtual screening. Virtual screening allows you to check that interaction between your ligand and your protein. So using molecular docking specifically. And of course, you can improve your molecular docking by, of course, doing molecular dynamics. Molecular dynamics gives motion, of course. Look, I can get this to play. Gives motion to your molecules and allows them to get a better inference from your docking, as it were. But what I want to draw your attention to here is that I know we have a lot of free software for docking, but you can also run some of this, especially if you have a large set of compounds that you want to run through your assay, your virtual screening. You can use the Galaxy Europe platform. Someone that Peter that will be speaking after me introduced to me, Chris Barnett, also have done quite a lot of work creating an African instance for Galaxy, specifically targeting computational chemistry. And he's a very nice young man, and they've done quite a lot of work on that. Okay, so I just want to mention a couple of case studies just before I wrap up. If I exit the time, so just tell me so I can stop. So are you thinking of case studies that combine all this together? The first one I want to talk about is something that I did a couple of years ago in Novartis, where we were trying to look for a cross-staged gene that is available at all the stages of Plasmodium's life cycle. And one of the databases that we use is PlasmoDB. PlasmoDB is well curated and always updated. And using PlasmoDB, we were able to find genes that are expressed in all the stages of the Plasmodium life cycle. And from there, we, using a couple of tools, identified those genes and located the proteins and did some screening, virtual screening against those proteins and identified some compounds which we tested. And we were able to find some highly active compounds. So this is one where we knew the pathophysiology, explored the possible targets, and did virtual screening. At the beginning of the COVID-19 pandemic, some guys in CSIR in South Africa approached me. They wanted to make a mask that is able to trap the coronavirus. So what I did for them was to look at the design for the map, for the mask, and check how it interacts with the spike protein or the membrane of the virus. And we want to be able to design a specific pegylated, as it were, polymer that can trap the virus. So we did that, and they are currently manufacturing that. Another thing that I worked with is assisting a group here that is working on nanotechnology. So we know that within the macrophage, the TB seems to enter some sort of a resting stage. So what they wanted to do was to stimulate the macrophage so that they can respond or kind of identify or wake up, as it were, to the TB that is present. So they wanted to stimulate a particular protein called dectin. And what they do that was to design a PEG nanoparticle pegylated with Cortland. So what they weren't sure the monomers, the amount of monomers they should use. So I did a simulation for them to tell them the monomers that will be available, that will be sufficient to stimulate a dectin, and they use that to form the nanoparticles. And we published that this year. They were able to use that to stimulate the macrophages. This is an example of using molecular docking or virtual screening. The interaction between your spike protein of the coronavirus and the human receptor. And what we did was to explore possible drugs that could interfere with that interaction. And we also looked at the interaction themselves, that this is linked to what I said yesterday when Chinwe was speaking, really understanding what that interaction is. And recently, South Africa mentioned that there's another variant that has been identified. And we want to be able to understand how that new variant or the different variants interact with the receptor in order to gain access to the cell. So we're using molecular dynamics. Like I said yesterday, I'm hoping to use topology, like she mentioned, to explore that. Another thing that I'm working with Dr. Moses and the group is to look at mutants from a particular organism and trying to see to really understand why the organism is actually mutating a particular protein and how a particular drug that was initially active, no longer active. So what we did here was to look at the pharmacophore modeling of the interaction of that compound with the various mutants. And you could see from here, just by running this modeling, I could see that the same compound interacting with different mutants have different pharmacophore in its interaction with different mutants. So for those that are doing molecular docking, just taking it a step into reliability, MMGBSA or QMM is another technique that is being used now to improve the prediction for molecular docking. And so basically what that does is that, so these guys came to me, they found, they tried to design compound against this target in Plasmodium, and they found a particular compound using their docking that works very well. But in their in vitro assay, there was no correlation between what they saw in the docking and that. So what I did was to apply MMGBSA rescoring, now actually sulfating the system and doing that rescoring based on that. And we were able to now rescore the docking output. And I said, okay, what you guys got was actually wrong. This compound will more likely be active. And when they repeated the assay, they found out that the rescoring actually predicted the most active compound. Okay, now in terms of data analytics and machine learning, we previously, a couple of years ago, published a paper looking at models that can identify anti-malaria compounds from natural products. Recently, my final year student here did a project to look at machine learning models for angiotensin converting enzyme inhibitors. So the wonderful thing about this is that I couldn't see them because we had to work remotely, but I could point them to a platform to use. So Data Robots, although it's not free, because we moaned that we are from Africa and the rest of them, they did allow us to use it, and they allows us to say we can publish the result from using this platform. So it's an automated machine learning platform. it gives a very easy way for the student to actually understand the modeling that they did, because they could interact, a very nice interactive platform. You don't need to code anything, but you still need to understand what you're doing in this. So this platform, if you moan long enough, you might be able to get something out of that. And finally, de novo drug synthesis is one area that I'm interested in, designing what we call an evolutionary algorithm that's able to use a parent compound that you know is active, and you want to create analogs from that compound, but you want your analog to fit a particular characteristic. So you can pre-design those characteristics. I want my compound to be like this number of hydrogen bond, this number of hydrogen bond donor, this amount of, in terms of weight. And by that, setting those criteria, you put it into that algorithm, is able to take that parent's compound and design generations from that. And you can now filter those generations based on the scoring of fitness to purpose. You can now select those generations and test them. So we've done this with a master's student, and we were able to show that the output from this did have the desired activity that we wanted. So in conclusion, I just want to say that we need to keep developing capacity in omics, and I want to appreciate the organizers for organizing this. I'm very passionate about developing capacity, but I think what we need to also do is to use available resources, the databases and the tools that are already out there. I hope that I've been able to achieve my aim to, at the end of the day here, make somebody to be able to sit with their laptop and internet and be able to say, I want to design a study and execute it and see what it is that I can get out of that. But we also need to increase computational capacity. I haven't said that. There will be things that you cannot run on your own, but there are web servers that you can also use. At Galaxy, Peter will tell you, you've done a lot of work on Galaxy. And of course, we need to also prioritize investment in human and computational infrastructure. Finally, I think we need to continue to reach out to the industry and see how we can solve problems that are that they need that they have in order for us to continue to be relevant. Thank you so much. I acknowledge all that have supported me along the way, and of course, appreciate the organizers. Thank you.
All right. Thank you so much for that beautiful presentation, Dr. Samuel. It was highly insightful, and I've learned a lot. Also, we now go to the Q&A section, and the floor is now open for question and answer. If anybody have a questions, you can just please type into the chat so that I can read it out.
Maybe I can start.
Okay.
Just to know, Dr. Samuel, the mask, I mean, that you are able to develop in such a way that it could trap coronaviruses. Could you confirm if there's a paper on that already?
Okay, so at this point, I'm not sure there is a paper, but there was a presentation. I think I put on my slide. I can share that. I'm not sure. So I know it was presented at the Chemical Association of American Chemical. Let me see. Yeah, I know it was presented at the conference, but the paper is not yet out. Issues around patency, I think that's the major issue. So yeah, but I know they've tested the first batch of this mask.
Did you consider, I mean, the saturation of the mask, maybe at a particular point, maybe if the person wearing the mask is already infected with the coronavirus? Do you think that at a point, maybe as you continue talking with the mask, maybe the chemistry of the whole thing might be affected and the transmission could continue? Okay, so the idea is to prevent it from coming into the the mouth. Yeah. That was the design. So the polymer was designed such a way that they tried different hydrophobic modification of the polymer, before they built the network. And they tried that, and my job really was to see, based on those networks,
which one of them is able to prevent the virus from passing through.
All right. Okay, thank you for answering this question. We have a couple of questions in the chat, and then I will go the one from Adepawale Alade said that what is the name of the drug platform for the de novo drug design?
Okay,
that's question one. I think she has two. Let me maybe just take the two there. And then he said, is SBV interoperable with ChemDB, ChemBL? ChemBL, yeah.
Okay, I didn't get that second one.
It's already in the chat.
So I'm trying to locate the what's the name of the person?
Adebowale Alade.
Okay.
Yeah, you can just scroll up, you see the two questions there.
Oh, okay, I see now.
Yeah.
Okay, is SBV interoperable with Campbell? Okay. Okay, what are you referring to as SBV? I hope you are speaking the same language.
Okay, we just go ahead and answer the first one if you have that.
Okay, it's okay. There's so the de novo platform is actually one of the platforms that we introduced at a particular training. We are still going to have a training, Dr. Moses, I believe, next year. So I think one of the things we'll do is to introduce you to those to that platform. At this point, I might not be able to share the algorithm that runs on that, but in the nearest future, we'll share the algorithm that uses that evolutionary algorithm to run that process. Yeah, that's. I don't understand the second question.
Okay. So, okay, then the same person also is asking that in addition to Zinc 15, one can download. Can one download correctable compound from PubChem or ChemSpider? Yeah.
Yeah, I know PubChem is free. I don't know about ChemSpider, but it's free. I'm not sure.
Okay.
PubChem is free. Campbell is also free. Yeah.
Okay, then another one says,
I see the SBV you're talking about now. All right. Okay, so the SBV improver it's not free. So what I've spoken to them. We are still talking. Hopefully they will recognize the fact that I'm from Africa and give me some access to their platform. Yeah, but they know they've collected data from Campbell, from PubChem, from different areas to run that machine learning platform. Yeah.
Okay, and then I think another question here says, what software did you use at the ground level of your de novo drug design? Okay,
yes, I said we're going to introduce that, especially for those that have some sort of knowledge in Python coding. Yeah, we can, but it's not. So it's a generic. It's a combination of some codes that I put together. But yeah, we'll talk about that.
Okay, so what you're saying is that's not it's not a software, it's a code you wrote.
Yeah, because there's a software, but there's a code that goes with it.
With the software, okay, to run.
Yeah, allows you to do that.
Okay. I think I got you. So let's go.
Yeah, I think that's about it.
There's still more questions here.
Okay.
You said you mentioned database for communicable disease. Is there anyone for non-communicable diseases?
I'm sure there is. There will be a lot of databases for non-communicable diseases. I mean, Professor Shegun spoke yesterday, and I took a note. Immediately he left, he finished, I checked. I can't remember the website now, but I did check immediately, and I saw some of the studies that he mentioned. You remember I asked him, where can we get the data for this presentation? And I quickly checked it, and I found that. So there are databases.
Okay. Yeah. All right. Okay, so due to the time, we need to move down to the next speaker because we don't have much time because there's a couple of questions.
Thank you so much.
You can just email your questions or you just send your question. Do you have an email that you can direct their question to so that you can just drop it in the chat? Anyone that still wants to go for further inquiry can still reach.
The consortium email, of course, most everybody here should have been receiving email from that.
Okay, so
send their questions back to that and we'll take care of this.
Okay, all right. So thank you so much. We really appreciate your time, Dr. Sam. we wish you success in your future research. Now, if we go to the next speaker, which is Peter Hildesing. I have my apology if I called that wrongly. Peter is a bioinformatician with a computer science and computer system infrastructure background. His focus is on SAMBI. He's building the tools to enhance genomic surveillance of some of the world's most deadly pathogens like tuberculosis and SARS-CoV-2. And his research is focusing on translating bioinformatics from research to accessible, reproducible workflow applicable to public health needs. In addition to bioinformatics and software engineering, he collaborates with colleagues in Africa CDC and WHO African regional offices to provide training and support for bioinformatics and genomic surveillance in the public health context. His dream is to aid in stopping outbreaks before they become epidemics and to grow the African capacity to do so. Peter, I think the floor is yours. You can take it right away.
Thank you so much. I trust that you can see my slides.
Yeah, we can. Good.
Let me take it away. Okay, so yeah, just a little bit of my personal journey. Thank you, Sam, for mentioning some of my interests. So this is SANBI at the University of the Western Cape. We are here on the top floor of the Life Sciences building of our university. And my background, as was said, is computer science, which I studied at the University of Cape Town. And then in 1996, I moved to University of the Western Cape, originally merely to be the systems administrator for the newly formed South African National Bioinformatics Institute. And being there for some time and having students ask me a lot of questions about computers, I had to start understanding what they meant about biology. And therefore, I migrated over time towards bioinformatics, where I now lead a software development team and am an emerging academic in the field of bioinformatics. And then a few years ago, we started collaborating with the Africa Centers for Disease Control and Prevention and also the WHO Regional Office for Africa. So in fact, if you do visit our offices these days, you will see proudly displayed the Regional Center of Excellence for Genomic Surveillance and Bioinformatics, which is a project that we are developing with WHO Afro at the moment. This Galaxy software was mentioned. I think people can Google it and follow the link in my slides when they get them online. It is, to me, a quite user-friendly bioinformatics platform, and very importantly, is a platform that is open and allows us as engineers and as trainers to collaborate on a shared project that can then be developed for all. It can be used in chemoinformatics more recently, but I mostly use it for biological sequence analysis, and particularly that of pathogens. And in fact, I'm sure people by now have heard of the new variant discovered in South Africa, the SARS-CoV-2 B11529. The first samples of that processed in South Africa were from the Iron Torrent sequencing platform, and they were processed on a Galaxy server run by the ISS called Ares in Italy, because they had a pipeline available for processing Iron Torrent data. And that just shows what such accessible platforms can do. They can allow people around the world to collaborate very rapidly. Okay, but enough about me. I want to talk about our continent, which, as I say, I feel it's a continent besieged both by the legacies and ongoing influence of colonialism and imperialism, and also by a three-part epidemics of poverty, inequality, and infectious diseases. So here we have a map of the GDP per capita of African countries. We can see that, unfortunately, some of our most populous countries, and in fact resource-rich countries on the continent, are are suffering terrible poverty. But we also see that even where we have some relative wealth in the continent, this is a map of the so-called Gini coefficient. We have high degrees of inequality. And anybody who works in the field of public health will know that diseases stalk poverty and inequality. This is the weekly map of outbreaks, disease outbreaks on the African continent, and 132. Obviously, we have SARS-CoV-2 all over the place, but we have ongoing pandemics of HIV, tuberculosis, and we have epidemics of diseases such as monkeypox that when they show up in the United States, everybody runs around and gets very excited. And here we seem to just live with these things as if it's inevitable. One of the problems is that you can't fight what you can't see, because I'm speaking to a Nigerian audience, and my surname is Van Hersden, by the way, but I'm quite happy for it to be massacred, because unfortunately my colleagues from Nigeria suffer the same thing to their names on a regular basis. So I wanted to look at what's going on in terms of the outbreaks that are listed in Nigeria. And the thing that I want to highlight is not so much the absolute number of cases, but what is the relationship between the number of cases and the number of cases that are diagnostically confirmed. So some priority diseases like circulating vaccine-derived polio, there is focused diagnostics on it. I haven't included the SARS-CoV-2, but effectively in these outbreak reports, every single case of SARS-CoV-2 is confirmed because it's kind of a bit circular. We only define the cases by the diagnostic, but we see a gap in diseases like monkeypox, where we have suspected cases, but a smaller number are confirmed. Okay. And this is despite the best efforts of the Nigeria Centers for Disease Control, which I know has been growing quite amazingly over the last few years. A much more troubling picture we find in the Democratic Republic of Congo, where, again, looking at monkeypox as a disease, look at the number of case reports, and the number, only 39 out of 9,000 were actually confirmed. This is a disease with a case fatality rate of about 3% at the moment, according to the current estimates. And plague, a plague outbreak where there's not a single laboratory confirmed case. What really got to me was the recent meningitis outbreak. And not only are there a very small number of confirmed cases, compared to the total number of cases, but the time from the outbreak being detected to the causative agent being positively identified was more than a month, and eventually that identification happened at Institut Pasteur in Paris. And if you might know that one of the major causes of meningitis in our people on the continent is Neisseria meningitis, but then you need to know which serogroup it is, because this is a vaccine-preventable disease, but you need to deploy these vaccines rapidly and of the correct type. So how do you do this kind of confirmation rapidly? And that means that it's not only important to know what disease we have on the continent, but what type exactly, and what are the genomic dynamics, what are the epidemiological dynamics of these diseases. By the way, the polyvalent meningitis vaccine at the moment is about $30 per dose in the United States. So this is absolutely unaffordable for us. So these are all connected: strengthening health systems, strengthening access to medical products, and strengthening diagnostics. So let's have a look. This is a slide I put together, also together with the Public Health Alliance for Genomic Epidemiology, on how we start looking at diagnostics in the world of SARS-CoV-2, and diagnostics and ultimately genomics. So we start with a number of different systems. So we've got healthcare workers at the coalface collecting data, collecting patient data, but the patient data is going one way into the some form of storage of patient and case information now that is often on paper, but it is starting to become more electronic in some areas. And then that goes in another direction towards the laboratory, where it gets a sample ID, hopefully a barcode if you've got that, and then stored in a particular freezer, where an epidemiologist is coming up with some kind of strategy to select samples for deeper epidemiological monitoring, and in the case that I'm going to be describing, genomic sequencing. Although, as I said, very unevenly on the continent. Once you've got the sample from the freezer for bioinformatics, we have a whole pipeline, and I'm not talking about the pipelines today, that's a different discussion, but for every pathogen, there's a bunch of reference data that we rely on. Now, obviously the SARS-CoV-2 reference genome, information on possible contaminants that we might want to remove, the primer schemes, but then also this thing called pangolin and nextclade. So we are relying on a global database of prevalence of these different lineages. But this is true not only for SARS-CoV-2, but for any other pathogen that we're facing. So when we had a listeriosis outbreak, a rather large one in 2017, 2018 in South Africa, then in terms of the so-called MLST databases, the core genome and whole genome MLST databases, we did not have a locally maintained database of the diversity of listeria pathogens in our region, and we were relying on Institut Pasteur to do the detailed typing of the pathogen. So developing capacity means not only developing the capacity to run these pipelines, but it also means developing this long-term knowledge in the form of databases that we both use and can need to contribute to. Then once we've got it at the moment, this is a picture of approximately how things happen at the moment. That most of the sequencing on the continent has its origins, the pathogen sequencing has its origins in the academic sector, and I'll describe a little bit why that is later. It starts from, this sequencing is sitting here in this little box. But remember, we're starting with samples that start with the healthcare workers, and the data for that is going into a health data center, either private sector or public sector. And then we have to draw that data to actually analyze the meaning of the sequence results, to actually make sense of them. We have to bring the data back together. And a lot of the systems for doing this at the moment are ad hoc and somewhat slow. Once the results are published, mostly there is no distinction between data sharing and data publishing at the moment. In other words, the sequences are being put into a database like GISAID, and then when people want to do larger analysis, they draw on GISAID. And in fact, yesterday, a colleague from the UK contacted me, say, look, I've just seen these new things in GISAID. This was just a few hours before the press conference talking about the new variant. What's going on here? And this fact that there's no distinction between publishing and sharing means that countries can get quite sensitive about putting data out like this. We saw last night that making the data about the new variant of SARS-CoV-2 available immediately led to travel bans against Southern African countries from the UK and Israel. So this system is what I call scattered sequencing. It's all like systems that are kind of mashed together, but they're not really designed around a genomic surveillance perspective. Also, the priority for a lot of the groups that have been involved in sequencing has been to get research insights, so preprints and papers out there, rather than developing dashboards that are actually feeding back into the public health world. That is starting to change a little bit, but it has really been a problem in terms of feeding back into the system that can deal with disease outbreaks. What we are hoping to see is an integrating system with multiple groups using a common platform to work on data according to policies that are agreed by the relevant institutional actors, and that can then share tools, dashboards, data sharing infrastructure, and then have a rapid turnaround, both integrating the health contextual data and working on the data on the shared platform, so that we can build a better platform for genomic surveillance on the African continent. I mean, as I mentioned Galaxy before, and I think that has a great role to play here, but there's a lot of work that needs to happen in terms of the data sharing and data storage platforms, and then of course capacity building and accessibility. I mentioned earlier that a lot of what has happened has grown from the academic sector. And if you recall the picture also of wealth and inequality on the African continent, then this diagram, which is now, of course, somewhat outdated on the number of next generation sequencing devices on the African continent, should be not that surprising. The majority in South Africa, some in Kenya, some in Nigeria, and then some in North Africa, Egypt, Morocco, etc. And these generally correspond to where sequences have been published on the African continent. And for a long, up until now, a lot of these devices have been funded by academic funders and sitting in research settings rather than in public health settings. That also kind of corresponds approximately to the number of sequences that are deposited in this GISA database. This is, as of this morning, it's not entirely up to date, but let's call it, the last week's figures. And we see that the groups that are doing most depositing of sequence data in South Africa, the National Institute for Communicable Diseases, CRISP, and its successor organization, SERI. But then in Kenya, the Kemri Wellcome Trust. This is another South African group at UCT, South African group again. And then in Nigeria, the African Center of Excellence for Genomics of Infectious Diseases, SGID. So we see that this majority of work, as we saw in this previous diagram, where the sequences are in South Africa, but also some from Kenya, some from Nigeria, but really drawing on the groups who have a history of being academic groups with funding via academic channels. One of the challenges we're seeing is how long does it take between sample collection and the sample being available for as data? And so we see here in South Africa that it is, while we got incredible turnaround times with the recent samples, the majority of samples have taken approximately a month to become available. In Nigeria, it's a bit longer, almost 50 days. But these things are strongly bimodal. So we see sometimes people going back and collecting samples from quite some time back to fill in the previous history of the pandemic. So we see these kind of bumps over here. So the poorer countries have a long smear of time from sample collection to sequencing available. This is DRC over here, Zimbabwe over there. So what can we do to change this? Okay, one of the crucial elements is human capacity development, and this is a particular bugbear of mine. As I mentioned, I started at SANBI as the lab assistant administrator. That was basically me down here. Okay, I was formally trained on this academic level over here. Effectively, I learned a lot of what I knew on the job, and thankfully, I grew along with SANBI. But right now, if you want to bring people in to build new tools for genomic surveillance, for maintaining the computational infrastructure that you require to do genomic surveillance, there is very little of a pathway into these fields directly within our human capital development system. Also because, as we look here, this is specialized research computing, which is what is giving us the tools for genomic epidemiology. but it relies on the more broader scientific computing and then ultimately general purpose computing. By the time you're getting down here, you're competing with industry for your staffing pool directly, and therefore retaining these people and giving them a pathway to growth within our institutions is a significant challenge. And it's not something that our managers necessarily and our leaders are necessarily understanding, that when in the academic sector, there's a pathway towards advancement, and as a student or as a faculty member within our public health institutions, often there is some overlap between that pathway and the academic pathway. But in terms of just these kind of more technical roles, they often are not, one can stay in the same job category for quite some time. Then in terms of institutional collaboration, right now we have to have collaboration because in fact, neither side can go it alone. To be honest, the National Public Health Institutes often have the samples, and the academia often has students and sometimes faculty members with skills. Okay, so what are the incentives for collaboration? Unfortunately, these are sometimes not balanced incentives, because the job of a National Public Health Institute is to provide a service, an ongoing service that must be reliable and routine. So processing the samples all the time, providing the reports to decision makers on a regular basis. The academia, you're being measured in terms of publication output, in terms of students produced, in terms of the teaching that you are doing, and there's some flexibility there. And in fact, students have played an incredible role during this pandemic, and in fact, students have contributed hugely towards this field of genomic surveillance. The pangolin tool that I mentioned earlier, that gives us names like B.1.1.529, was maintained during most of last year by a couple of PhD students in Edinburgh, one of whom has just finally graduated. So I think it's amazing that she could finish her thesis while running the tool as well. HGID, their main training and bioinformatics workforce is a couple of postdoc and PhD students. Again, amazing people. And they've got this flexibility. And the same thing happened with Seri, and to some extent with us at Sanbi, that when the job had to shift, then the flexibility of academia allowed these groups to shift very rapidly. Whereas, when you are in a public health lab, the stream of regular work doesn't slow down for you to take on a new task, okay? Then what are institutional structures? How do I even find somebody at the institution to collaborate with? Will they talk to me? Will there be professional jealousy or so on? And of course, what egos are involved? And then what are the outputs, okay? So are we producing surveillance reports? Are we producing papers? How can we satisfy the different needs? And then this all comes down to who benefits. As an individual, career development, those kind of things. The community, who are we serving? And what are they getting out of the work that we're doing? Okay, so is it just basically, getting a bunch of geeks jobs, or is it actually servicing our communities who are at the receiving end of these diseases? The institutions that we belong to, how do our leaders higher up in institutions understand our work and support our work? And then we have to think beyond country, region, and continent. So yesterday we had a press conference about this new variant in South Africa. The UK media was already all over the place because of the early reports of it. I have not heard one person ask, what is the perspective of Botswana? Remember, the first 3 samples of this new variant were sequenced in Botswana. What is the perspective of Botswana, Namibia, Zimbabwe, Mozambique, Angola, so on? The last time we got a new variant of concern in South Africa, the so-called beta variant, it really hit our neighbors all the way up in Southern Africa, up to Malawi. The second wave, so-called, or the beta wave was really, really intense. So we cannot take a single country perspective. We have to think regionally. We have to think as a continent. And in fact, the African Centers for Disease Control and the African Union has been incredible during this pandemic. But this is not just about SARS-CoV-2. What are we thinking about other diseases? Okay, other collaborations. And then, as I mentioned, continental partnerships, the World Health Organization for Africa, which has many, many responsibilities. And this is one of the things that is struggling, is that we know that we got many diseases happening at the same time. Okay, so and the Africa CDC, if you look into the Africa CDC framework from, I think it was 2018 or 2019, for strengthening national public health institutions, this has really given us a lot of the framework for what we are now fitting genomic surveillance into. Okay, and then the Centers of Excellence, the African Centers of Excellence for Genomics of Infectious Diseases, ASGID in Ede, SANBI in South Africa, SERI, which is now in Stellenbosch, South Africa. And I also wanted to give a nod to KEMRI in Kenya. These are some, but not all of the resources that we have on the African continent. But again, how can we get them working together? And none of these are working exactly in the same organization, because we all have our own responsibilities. So, how can we actually use this to build capacity development that is looking at beyond the individual institution? Okay. Part of this comes, of course, down to funding, and the funding landscape, as I wanted to illustrate on this slide, it is a landscape dominated by agencies outside of the African continent. So from the US CDC and the NIH, I will note that one of these is a public health oriented agency, and the other one is a research-oriented agency.
Sorry to cut you in.
Yeah.
You have about 2 minutes to go, because
2 minutes. Sorry. Okay, I will finish. I'm just about at the end of my slides. Okay. In Francophone Africa, the Institute Pasteur has a huge footprint. We know in the Anglophone part of the continent, some countries we have the Wellcome Trust, and now more recently the Rockefeller Foundation is coming in with its funding. How does this this funding is great in terms of developing institutions like some of the ones mentioned on the previous slides, but how does it relate to African goals? Okay, and where are the significant funding from our own continent? Finally, I just wanted to speak to the need for innovation. So what is our innovation policy on this continent? So do we only think of innovation as a path to market? And where are we sitting with training and training materials? Because what I find is I'm doing training, maybe Sam will do training and Abdullah will do training or something like that, but we are doing these things in silos. So how are we actually developing training and training materials in a collaborative way? What models of staff development can we share and learn from together? And then, are collaboration and innovation working together, or are they working at odds? Do we see the intellectual property landscape and the individual advancement landscape as actually undermining the collaboration that we need on the African continent? Sorry, I had to talk rather fast there at the end, but now is the time to seize the moment. We must build networks. All the world is not SARS-CoV-2. There are other diseases out there. We have to figure out how to be humble in our fields and how to share the benefits in an equitable way as we grow in this world of genomic epidemiology. And I will leave it there, and hopefully there will be a minute or so for some questions. Thank you.
All right. Thank you so much for the presentation. I think the floor will be open now for questions. But before that, let me just go first. I want to know the new variant that was just identified in South Africa or Botswana. Would it have any effect on the existing vaccine? Because since we know the mRNA vaccine was designed based on the genomic sequence, so now that we have a new one, will it render the existing vaccine impotent, or what do we see to that?
Okay, so the first thing I can say is that I can almost entirely guarantee you. Okay, So do you want to line up a few questions and I answer them together?
Yeah. Okay. Yeah. This question just from me. The second question I have is when you, I guess, since as a bioinformatician, you know what they call like the third generation sequencer, which is like the nanopore and then PacBio. So now, because when it comes to identification of those, because you can actually map the entire genome of the SARS-CoV-2 at once, as where you compare to other short sequence like Illumina or MiSeq. So what technology are they using in Africa? Are they using Illumina or they are using the nanopore, the third generation sequencer platform for their sequencing?
Okay, so let me start with the second one first because it's quite easier. The majority of sequences in South Africa have been done, and that is at the moment the majority of the continent have been done on the Illumina platform, because even though the reads are short, the genome variation in SARS-CoV-2 is not so large that that it causes a problem for short reads. Okay, but what we're seeing is a widespread adoption of the nanopore platform, because while the nanopore platform has its problems with accuracy, although those are getting resolved now at the moment, the new chemistry is really quite impressive. The capital investment in a nanopore sequencer is much lower. Okay, so we're seeing these things being deployed in institutions across the African continent. They're quite easy to get up and going. But it always does still hit somewhat of a bottleneck when it comes to bioinformatics and the software analysis. In fact, I mean, a lot of the bioinformatics is still command line oriented, and that's a skill that's quite rare on the continent. So the sequencing, it has to all feed into a bioinformatics development program. But yeah, I would say that Illumina is still the majority of sequences, but with nanopore rapidly gaining.
Because I'm looking at it from the point of, because you have to ship some of it. Like for example, maybe you collect a sample from Nigeria, Congo, and you have to transfer your sample to other places before you're sequencing. So that could actually introduce bias. But with nanopore, you can actually go to the site. In the site you do your sequencing there, and after doing your sequencing, then you can actually send your data to whoever. And the one advantage is that you could actually multiplex on nanopore, because
96 samples, yes.
Yeah, you can actually multiplex. So that would be an area, but it might be a bit expensive. But at the long run, the accuracy of your data might be at least worthwhile. I don't know, because of some site operations.
You know, we can talk a long time about the technical details here, but yes, look, I think nanopore is great possibility. Also with the new Mark 1C devices, there's a possibility to even run some bioinformatics software on the actual nanopore sequencer platform. And I think there's great possibilities. One thing that worries me is that we've seen what the consequences of relying on a few Western companies for vaccines and diagnostics is. Are we going to end up in a bottleneck where we are at the mercy of these companies for the supplies for our sequencers? So if there is a need for kits, now we might not be able to get the kits because labs in the north or in the global north are buying them. So it's something that I always keep an eye on the availability of medical supplies and what it means in terms of the single supplier problem. But on the question of the new variant, okay, firstly, we need to look at the immune system on two levels. Okay, it's not only antibodies, it's also T cell response, B cell response, and so on and so forth. Okay, we know from previous research on similar mutations in this virus that we might see a drop in the antibody efficiency, but and therefore some loss of protection against infection, but the T cells tend to stay quite good no matter the different variants that we've seen. So I don't believe that I believe that vaccines continue to be our number one tool against this disease, including this new variant. but we will very rapidly, right now they are taking this thing into the lab to do antibody assays. It would take some time longer to do T-cell assays. So I think that it is worrying because one of the kind of walls against the disease is the immunity from both vaccines and prior infection. And if that wall starts crumbling, it is worrying in terms of the scale of the next outbreaks. But I still believe that in terms of severe disease, that is the vaccines are likely to still work.
Okay, all right. Thank you for that. Do we have any more questions from the floor? You can just chat, you can type it in.
Yeah, I just want to thank Peter for this, his passion for this and his nice presentation. Maybe from one of your slides, I could see that you mentioned that Nigeria has many platforms for, I think, like 20 devices already. So I don't know if you have idea of the distribution of these sequencing platforms in Nigeria, because I think we know few, but I don't know if you know more from there.
Well, I mean, even this year, there's the sequencing at University of Ibadan. NIMR has the Chinese MTI sequencer. Of course, a lot of sequencing at HGID. Then there is some nanopore capacity at NCDC in Abuja, and nanocore capacity at, I think, University of Jos. Although I know America is always struggling to get some reagents for that thing. And I cannot say off the top of my head where the other sequences are, but those are the ones that I know of, just thinking quickly. So as I said, the centers in Nigeria also have been academic historically, HGID, and now Prof. Iruka Okeke's group at Ibadan, but there is some capacity also in the more health-oriented NIMRs and in NCDC. I don't know where the rest are that don't count in that number, but yeah.
Okay. We'll take one more last question, Dr. Samuel. Can you unmute yourself and ask a question?
Yeah. I actually typed it on the chat, but
okay.
In terms of the workflow, that you spoke about. So I'm looking at the data coming up from the clinic and the research in terms of the sequencing and merging them at the end. I'm thinking until the health workers really see the role of genomic data in the management of diseases, we're still going to have this parallel lines. So I'm thinking, who is doing the advocacy to the healthcare workers in terms of getting them to see the role of this output from the sequencing and really now merging that into, because I know in South Africa, we change the treatment guidelines quite often, but we still don't see a reflection of the output from such genomic surveillance.
Yeah, and I mean, I think who and where, right? Because I mean, I think it's a question of basically how these things get adopted. For instance, in South Africa, we have a very well-funded private healthcare system, but it's really locked down by companies in terms of getting access to innovation there. There's actually not a lot of innovation happening in that space. But part of the problem has always been the fact that you can get 80% and then 20%, right? So we've seen emergency responses. In fact, the South African use of whole genome sequencing for the listeriosis outbreak was an emergency response because making things available and easy, that last 20% on top of the 80% has been the challenge in terms of translation, because you can go to a healthcare worker and say, we've got this thing, but until you actually show it in a way that it is meaningful to them in a language that they understand, you're not going to make progress. However, what I said in my slide there towards the end of seizing the opportunity, we're seeing a major investment in bioinformatics and sequencing. Our National Center for Tuberculosis in South Africa is getting an in-house bioinformatician for the first time ever, for instance. The NICD in South Africa has increased its bioinformatics staffing by approximately 3 times during this pandemic. So I don't know what's going to happen in terms of ongoing funding. NCDC is also, shifting a lot in this direction. But we have the opportunity of the skilled people being there. I think what we now need to do is find these spaces for conversation. How can we create the spaces for conversation in a way that avoids the problems of ego and hubris that have ditched some conversation in the past, and then seize the moment and actually build a powerful network to actually show that this thing brings results.
Yeah, cool. And it doesn't have to take a pandemic for us to respond every time, but it is what
but we have had the pandemic, and that is we are out of time, I think.
We're out of time, so we need to move to the next presenter. So thank you so much.
Thank you, Peter. I appreciate it.
Thank you. Cheers.
We really appreciate your presentation. So we're moving next to our next presenter now, and that is in the person of Dr. Charles Oluwaseun Adetunji. I think if it's on, if you can just put in a slide. Dr. Charles Adetunji is presently a faculty member in the Department of Microbiology, Faculty of Science at Edo State University, Uzeni, Etsu, Edo State. It utilizes the application of biological techniques and microbial bioprocess for the actualization of sustainable development goals and agrarian revolution through quality teaching, research, and community development. He is currently the acting director of intellectual property and technology transfer, sub-dean for Faculty of Science, and the head of Department of Microbiology of ETSU. Pardon me. He is a visiting professor and the executive director for the Center of Biotechnology, Precious Cornerstone University, Ibadan. He has won several scientific awards and grants from renowned academic bodies like the Council of Science and Industrial Research, CSJR, India, Department of Biotechnology, DBT, India, and the World Academic of Science. He was ranked among the top 50 prolific authors in Nigeria between 2019 to date by ScienceVar and Scopus. His research interests include microbiology, biotechnology, post-harvest management, and nanotechnology. He was recently appointed as the president of the Chairman Governing Council of the Nigerian Bioinformatics and Genomic Network Society. He is presently a series editor with Taylors and Francis, USA, editing several textbooks on agriculture, biotechnology, nanotechnology, farm food, and environmental science. He has over the last 15 years, he has built strong working collaboration with reputable research group in numerous leading universities across the globe. He is a convener for the Recent Advances in Biotechnology, which is an annual international conference where renowned microbiologists and biotechnologists come together to share their latest discovery. He is the president and founder of Nigerian Post-Harvest and Food Biotechnology Society. Dr. Charles, you're welcome, and the floor is yours. You can take it right away. Dr. Adetunji? Can you also unmute him? I don't know if he's hearing us or we can't hear you. We can't hear you if you're speaking.
Charles, unmute yourself. I think you're muted.
Yeah.
Unmute yourself, Charles.
Can you hear us? Because
he could hear. I'm asking him to unmute. Okay, maybe I give him a call.
Okay, or maybe the host can just send me a request to tell him that you
I've done that also.
He said the host is not allowing him, so you can do that again. He said the host is not allowing the participant to unmute their mics.
Hello, good afternoon, everyone.
Yeah, we can hear you now.
Yeah, thanks very much. Sorry for that technical issues. So this morning, I'll be talking about new front ends in bioinformatics, the current scope of the ever-increasing population. So there is no need for me to make any introduction. Introduction has been made already. So I count it as a privilege for me to be here this morning to talk on new front ends in bioinformatics. As we know, the diverse part of the globe are going through a lot of challenges. Most especially, the whole world didn't and what happens to the whole world in the year 2019, the COVID-19. So, first of all, I want to thank God for the privilege, for the grace that we are alive for today, because that forms the basis of everything. Now, it has been stated that the whole globe is going to increase drastically to 9 billion in the year 2050, which shows there is a clarion call for all the scientists to work together. Now, it is now time for us to think of the best technology, how we could solve this global problem, global challenges. The human population increases every day, but we need to place a lot of techniques on ground for us to mitigate a lot of several diverse challenges. How do we use digital revolution to achieve sustainable development goal? How do we use digital revolution to solve the problem and maintain a sustainable system and community? How do we use informatics, bioinformatics to achieve a sustainable food, ocean and water, energy decarbonization, education, and a lot of stuff, most especially in this challenging time? So having said that, it has now become a point and a crucial clarion call on all the scientists to come together and work together and see how all these challenges could be resolved. Circular economy is a way in which we can harmonize all our ecosystem and restore diverse challenges. In order to do so, we need to use life cycle tool analysis to resolve several challenges, particularly several digital challenges in resolving global problems. Therefore, there is an urgent need to change circularity and sustainability for us to embrace diversity. We need to look into our education curriculum. This should be a kind of interdisciplinary foster. We all know bioinformatics. I want to have this in two perspectives. That is a kind of tools, techniques that support several kind of biological sciences, agricultural science, and a lot of other stuff, which is clearly scope to a new assay method and investigation method. It's a kind of science that supports a systematic analysis of two and investigation of a set of principle foundation of sort of diverse bioinformatics application. The backgrounds I'll be talking today encompass around computer information science, which form a need to understand tools, scientific approach, and need for research, biomedicine, agricultural science, and a lot of stuff. But we need to understand there are so many gaps before we could use this tool. One, biology students and researchers can't build their own tools. Computer science don't know what tools to build. Gap two, putting biology education and system method does not solve the problem. There are a lot of barriers like language, methodology, conceptualization. Another gap before we could solve the global problem. Computer science is a science of artificial, mainly concerned with human artifice, correlation limited made by conservation and mutilation. Biomedicine also. So that means with the help of bioinformatics, we could be able to overcome diverse challenges. We have the general tool, communications, database, analysis. So informatics has changed how science is being done. Now, we could divide informatics in terms of skill stuff. Know how we use the practical, causing cultural exchange, solving scientific problem, which means informatics is an information science. Now, if you look at my screens, this shows diverse pathway, but it depends on the approach we want to take. So it's like an interface. It's like a universal text. Before we could say, so where should we put emphasis from the perspective of new students and new scientists entering into this field, wondering about their career? Will bioinformatics turn out to be the main end product? Will it be a viable science or the right one? We will see. Now, before we could solve diverse challenges mitigating against advancements of mankind, we need to embrace sustainable development goal, which is an impetus over millennium development goal. So this will help us to design what particular solution, innovative solution, using biotechnology techniques. Now, we have a big question. Can bioinformatics whatever be a kind of tools that can help us to achieve sustainable development goal? So I would like to share some of my view experience as a bioinformatics and biotechnologist. I've talked about that the world population is going to increase drastically. What are we doing? My research cluster have been able to discover a new herbicide using a lot of bioinformatics too, because chemical herbicides have been a threat to farmers, and many of them could not avoid the cost of the highly cost of synthetic chemicide. So we came up with a bioherbicide that become a permanent replacement. Chemical herbicide is a kind of threat to food safety and ecosystem and human health. So we have used bioinformatics to identify the gene responsible for the production of a bioherbicide. We have deposited at SBR. We have shown the homology of different strains of isolate that could enhance farmer to farmer, and we have published it in a high impact factor. And we have also demonstrated the metabolomic aspect of it to establish the viable active components responsible for the bioherbicide activity. We went further to demonstrate the mode of action, how it works. So we have also established all the various active ingredients. We went further in order to establish, not using dry lab, we went further to the wet lab to establish the enzymatic activity and see how we could help the local farmers, because not all of them understand it. So we came up with a model, Animation Without Borders, that could explain how farmers that could not read English could explain all these bioinformatics tools in a simple language. Also, we've used locally available substrates to encourage farmers that could not use this technology. It has this stabilization. Now, we are talking about dry lab. The practical lab, dry lab, form a kind of roadmap to wet lab. So we went further to establish the ecosocial assessment of this newly discovered herbicide. We have established and we see that it's working and it's eco-friendly. Because anything that we have to consume, we must ensure the environmental safety, the health safety, and everything. We have also demonstrated it to the farmers. We have shown them how they could apply it. In terms of cost, it's cost feasible. We went further to document the patent in this regard, which have been approved by the federal government of Nigeria. We went further to see, through a collaboration with a scientist, Dr. Wadazani and Dr. Adetunji at University, we went further to see, can bioinformatics, nanotechnology, endophytes be a kind of means for delivery of active drug to restore several tropical diseases and challenges. So we have been able to establish some potential industrial strain that could produce drug, that could mitigate a high level of drug that have developed a lot of resistance. So this is joining the 3D structure, and with these are some of the isolates that we have isolated, we have characterized. Also, through a collaboration with Dr. Wadazani, we went further to see evolution and protein-based function of African mosaic cassava. Now, cassava is a kind of stable food that we consume mostly in Africa and in Nigeria. Africa contributes 60% of this to cassava production. It started as a stable food. But the yield loss was, in terms of yield, ranges between 1.9 to 2.7 billion. Now, we went to see, can the protein code of the virus, which has been established to have a multifunctional roles in the virus, have a kind of component that could mitigate this, and can it serve as a kind of pesticide against cassava mosaic disease? There are a lot of direct information on this, because this forms the basis of our justification for going to this. So we went further by using a lot of bioinformatics tools and bioinformatics techniques, and we have demonstrated the various codes. We have shown that this shows the distribution of conserved motif, and this is showing the various 3D of the protein isolate. This showing the biological process, the ontology, and all of them. Now, the study revealed that the protein codes could form a kind of pesticide that could be used in the management of this burial disease. And the MSA of the gene reveal high conserved domain of the 3D that run across the isolates, which could be used to indicate every primer for the disease of cassava mosaic virus. The gene association also predicts most of the protein to be associated with the viral capsids. The insight gained from this study also established that it could be explored for management of this virus. So this is showing the publication with Dr. Wadazani in this regard. Yeah, we went further to do a lot of profile SciComm of P450, which is ongoing, but because of time, I won't be able to go into many of them. And Dr. Wadazani, which is a member of the research group of our research cluster, have won an award in this regard also, showing a lot of SciComm P450 by award given to us by American Society of Microbiology Federation, European Microbiology Society, and supported by Melinda Gates Foundation. Also in our research group, we use crystal regularly interpret short part of the rapid a lot to achieve a lot of goals. As you know, this year award was won in these regards. So our research group won a research grant of recent through a kind of US, EU, African, Asian, Pacific, Caribbean Nano initiative. We are trying to see how we could blend the application of bioinformatics with nanotechnology. We discovered that when farmers apply most of their pesticide, it doesn't stay on plants, it went away. So we look at the way where we could allow biological components or active components to be delivered actively into the structure of plants, bearing in mind the hard cortical and the cell wall of plant. So this is showing the grant of 18 million for this particular project. So we went further to see could nanoparticle and CRISPR-19 Cas form a kind of delivery to solve several diseases. We have established that in our group. We went further, not only at the laboratory, because most of this technology we are talking about, we need to do rural and extension work. We form a synergy with essential workers. We have demonstrated at 6 geopolitical regions how most of this technology could be applied and tested. And we constantly have a kind of collaboration with Farmers Association and a lot of other forums. A lot of have been developed. In terms of environmental biotechnology, we've also established cool bioinformatics and informatics output help to maintain and ensure a smart city, a more cleaner environment. We have done a lot of work, and these are some of our population in this regard. And because of time, I might not be able to go deep. So what I want to bring out is that the application of bioinformatics has really been of help in order to achieve this. Now, there are a lot of plastics, a lot of contaminants in our environment. The application of bioinformatics has really been of help to achieve this. Now, during COVID-19, there are a lot of challenges that I've talked about. Now, this now shows that one of the most contagious diseases on earth that has ever affected mankind is COVID-19. Now, we as a bioinformatics and technologists, academicians, students, data science, needs to work together. How can we come up with our own vaccine? A lot of scientists, speakers have been talking about this, so I don't want to dwell more. So, but we need to develop a robust blueprint that need to have faith how we can ensure our diagnostic vaccine and therapeutic in order to cure this vaccine. There should be a blueprint. We should work together. We as a so during this COVID-19, we work together and see locally available resources, the bioresources. Because when you're talking about circular economy, we need to embrace resources, but we have not turned them into money. We need to borrow government government money. We need to borrow government money. So we as scientists need to harness a lot of several diverse bioresources. And one of them are available in Africa is presence of medicinal plants. So during this COVID-19, we went further to see history screening of some selected medicinal plants, and they could be a potent inhibitor of transferrin protease, inhibitor that could block the entry of COVID-19 into human cell. So we went further and we have established this using a lot of bioinformatics techniques, and this is showing some of the results. And this was done with a lot of Dr. Yedara from Mexico. So we were able to establish the 3D structure of the protein, the interaction between the residue of this particular protein and the plant active metabolites. And we see how it could inhibit COVID-19, and it has been established and it has been published. So CRISPR-19 has also been used in our lab for a lot of other things, but because of time, I won't want to waste time because I have a very short period of time. But presently, in our research group, we are using it for a lot of things. Now, there is a lot of projects we are doing presently now on biotechnology and health informatics. We are established how machine learning, artificial intelligence, deep learning, and a lot of other virtual argument in terms of mixed reality, virtual reality will help, and a lot of techniques how we can help cyber security issue in terms of biomedical, 3D, and a lot of other stuff we could help. Presently, some in our research cluster, we are using CRISPR to see how it will help to solve the problem of sickle cell anemia. Gene therapy have been established, and we are currently working on it. Also, we are seeing working on how it could help in mitigating several liver and kidney disease. And can it be a kind of solution to shortage of organ? We are using CRISPR to see that. This is not from our research group. Some scientists have also seen how CRISPR will help in mitigating malaria disease. So in reproductive medicine, it has been used to establish a lot of things. Out of gene therapy and CRISPR baby, a lot of work has been done in that regard. Also in non-viable human embryo, in preventing viral disease, scientists have established part of that. In fixing cancer disease, it has been established. In regulating a lot of hematological diseases, it has been established. So we can see CRISPR has been a very good bioinformatics tool that could help us to achieve a lot of challenges mitigating against achievement of sustainable level in terms of health delivery. Now, in order for me to round up, I would like to advise all of us that before we could achieve the goal of resolving cyber's global problem, whether in terms of agriculture, health, industry, food, we need to work together. We need to relate together and form a kind of interdisciplinary approach. This is the only way how we could solve the global problem. You could see it's not all about dry lab alone. Wet lab, farmers, policymaker, government needs to be incorporated into our work. Then we need to lobby with the government. I remember in the year 2016, when the agriculture was bound to Europe, I was the consultant invited by the Senate to come and address this global issue. So we need to talk with the government. We have the techniques and the scientists. We have what it takes, but we need to let them know what we are doing so that it will not be on our shelf. It will not be just ordinary thesis, just ordinary book. So we need to carry government along. So I would like to thank several professors that have been a great mentor. I've hosted in several of their labs, several vice chancellors, for the great privilege, several lab heads, a lot of, many of them for what their impact and a lot of other things. I'm very grateful for this golden opportunity. And I would like to invite as many as possible of us. I've been appointed as series editor, Bachelor of Science in US. There are a lot of books. I have more than 10,000 books that are in progress projects. So if anybody wants to collaborate, you are welcome. And I would like all of us to also join Nigerian Bioinformatics and Workworks. And you are welcome to join. And take home lesson. Blessed are thou that art hungered for righteousness, for they shall be filled. I want to appreciate the convener of this program, Dr. Moses Ikuna. Thanks so very much for inviting me as the keynote talk. On this note, I would like to say thank you, everyone, distinguished professors, Dr. Akiola, many other people on board. I'm very grateful for this great privilege. God bless you and remain blessed forever.
All right, thank you so much for that insightful presentation. It was an eye-opening, and I will see your great work you are doing in the science community. And now, I think the floor is now open for question and answer. Question to the presenter.
Yeah.
So I don't. Okay, do you want to go first, Moses?
Yeah, I have something in my mind. Though Charles was pretty fast in the entire presentation, but I still feel to ask one or two questions, Charles.
If anyone has a question, you can drop it in the chat box so I can read it out, or you can unmute yourself where you can ask.
Okay.
We still have some couple of minutes, like 5 minutes for questions. So
okay. Charles, thank you so much. I could see you do a lot of things at the same time. You know, many things: agriculture, biotechnology, HIV, SARS-CoV, and a lot of that. And I really appreciate your enthusiasm in things. My attention clicked on two issues. One is your the endophytes. You know, some endophyte studies you presented. Sorry, I could not get clearly what those endophytes are meant to do. Are they anti-tuberculosis? Are they anti-HIV? So it was not clear to me what is the final outcome of that research, maybe because of the speed.
Okay. Actually, sorry for my speed because I have a lot of slides, but I will go extensively deep with that. Endophytes are microorganisms that dwells in the inner tissue of plants, and they exhibit a kind of symbiosis relationship there. Now, some of these endophytes reside in the inner chamber of plants, and they have been established to produce a lot of useful metabolites. Now, what do? We use bioinformatics to identify a specific metabolite of interest being produced by this endophytic microorganism, whether bacteria or fungi or actinomycetes. So whenever we isolate these fungi, we do a lot of scaling up and production of the metabolite. We extract the active ingredient that constitute like a drug. Now, what we are now using is we use nanotechnology for targeted delivery of this drug against some challenges. But the first one we are doing right now is its anti-inflammatory effect. That is what we are doing presently, using this antibiotic. Now, we want to see also further, some of my PhD students are working further to see could this endophytic microorganism serve as a kind of anti-parasite, anti-malaria. So now we are demonstrating, establishing various biological activity of these nanodrugs produced from the metabolite of these endophytic microorganisms.
Okay, thank you.
All right. Okay, are you done? I have a couple of questions to ask.
Okay, I take the final one.
Okay.
Yeah, maybe on some compounds also that, or some plant compounds that affected the SARS-CoV membrane permeation and all of that. Again, it was not, what was the outcome eventually? Are there some potential compounds from these plants that currently is going through development, probably into anti-COVID drugs? And where are you in that research currently?
Okay, thank you very much. Actually, we'll be able to establish some of these phytochemicals that are responsible for the activity, biological activity against this COVID-19. Now, you know, it has to start from several procedures. We have done the computational aspect of it. Now we are trying to see how we could scale up and see how it could serve as a kind of vaccine. But you know, the process of vaccine is not just a day job. You need to start from the in silico aspect before you move further to that. So work is still ongoing, and there are a lot of several stages we have to go to before we could say we are going to the stage of vaccine.
Okay, thank you.
Thank you very much, sir.
Yeah, all right. Doctor, I have some couple of questions. So just on the same slide you are now, that same slide. I want to the plants you guys are working on, I guess you guys must have extracted the bioactive compound and know which one is exactly, or you're working with the root extract. That's my first question. My second question on that same one is what cells model are you guys using for the study? So because you said that you compound inhibit entry of SARS-CoV-2 into the cells. So what cells particularly were you guys using for that this thing? And then my final question goes to the CRISPR issue research on cell project. What are your subjects? Are you using human? Are you using CRISPR directly on human, or what are you using for your CRISPR study? What model are you using? Okay. Those are my three questions.
Okay, thank you very much. Actually, some of the active ingredients, we talk about active ingredients. The active ingredients that we isolated during this study are niacin, quercetin, and morazine, most especially from Zimba ovacille, moringa plants. I don't know, maybe I've answered that.
Okay, yeah. So those are also.
So what we actually were doing during this was we were trying to do a like of molecular docking to see maybe the potential inhibitor of transmembrane protein serine, which is an important protein, could form a kind of successful establishment with COVID-19 infection. Hello?
Okay, so
what we are doing, we are trying to establish a kind of binding affinity of the compounds using a kind of homology model of TMPRSS2 using molecular docking.
So if I get you right, you guys have not gotten to the stage of using a model or a cell model. It's just still in
No, sir. We are still, we are still on that process.
Because when we work more on in silico, most of the things that look so robust and look so sweet and so nice, they don't translate to the same thing when you bring them into the real model. So
yes,
that's my own this. And then for your CRISPR sickle cell, so is it still on in silico as well?
It's still on silico. We have not got it to advanced stage of it.
Okay, all right.
Yes,
thank you for that. Okay, thank you. And then I think somebody, the last one will pick here. I think I saw something on. Let me see. Okay, I think Ifeanyi Okuru was asking. Thank you, doctor. And he was asking that if you could throw more light on CRISPR. So I think that's a broad thing because if you start from CRISPR from now to the next 5 hours, we won't finish CRISPR. So I think we can just summarize the process.
Okay, actually, thank you so much, doctor. CRISPR is just like a scissors that you can use to trim specific gene of interest for you to achieve a particular goal in your informatics or computational biological science. So it's like a scissors. You must definitely have a particular gene of interest or whatever you are targeting. So it's like a scissors that you use to cut a particular kind of gene of interest that you want to use in your computational or biological activity or assay. It's a very big feat that if I begin to expand, like Dr. Akinola said, you can bear me witness that we will not live here today. That's why I'm very fast for my lecture. But maybe when time permits, some other time, we'll go deeply on CRISPR.
Yeah, okay. Thank you so much. CRISPR is also like a tool for genetic manipulation to genetic engineering, in which, as I said, you use a scissors to cut and you take it into a new genome and then you integrate. Then you see the effect of that in a particular organism. Thank you so much for that wonderful time. Thank you for the presentation. We really learned a lot from you. And
thank you, sir.
success in your research and your future endeavors. And then you keep it up. Thank you so much.
Thank you, sir. Thank you, sir.
Yeah. So, all right, moving on. So we're moving on to our next speaker now. The next speaker is going to be Dr. Giselle. Dr. Giselle is a senior bioinformatician at the Institute of Biomedical Technologies, ITB, of the Italian Research Council. And is responsible for the bioinformatics unit at the International Institute of Tropical Agriculture, IIT Ibadan, Nigeria. He is trained as a molecular biologist and received his master's as well as doctorate degree from the Swiss Federal Institute of Technologies in Zurich, Switzerland. He did his postdoctoral research at California, that's Berkeley, USA, and at the Novartis Foundation in Basel, Switzerland. He worked as a bioinformatician since 2001, first in the pharmaceutical company Novartis SA in Basel, Switzerland, and then at ITB in Bari, Italy. He specialized in developing biological database and algorithms for next generation sequencing data analysis. His major interest is in the investigation of the genes regulation mechanism using NGS data from transcriptome, small RNA, and DNA methylation data. His studies are mainly concentrated on cassava, maize, Arabidopsis, and human. Now is a member of the executive board of the Global Bioinformatics Network, EMBNet, and heavily involved in training events for NGS data analysis all over the world. Dr. Giselle, thank you, and you're welcome to take the floor. Can you, Dr. Adetuji, can you stop sharing? Or I don't know if you can stop sharing. Okay, all right. So Dr. Giselle, over to you. Take it up from there.
Maybe Tony could start. Maybe you can introduce Tony to start, then Giselle could come later.
Okay, Giselle is not available for now?
He's not currently online, so Tony could just start.
Okay, is Tony available?
Yeah, Tony is available.
And is ready? Okay,
yeah.
All right. So, if that's the case, then we'll go into our next speakers, which is Toyin. Toyin is actually a bioinformatician with a diverse background ranging from engineering, applied bioinformatics in plants research, and now applied bioinformatics in psychiatry. His current research is focusing on using multi-omic strategy that integrates gene expression, protein, DNA methylation, lipidomics, and SNP genotype and genotype data to predict aging-related outcomes and longevity. A part of his project aimed to develop a comprehensive framework for the discovery of genetic variants that explain variation in gene expression level, mostly expression quantitative trait, which is called EQTL in older adults. His result could be used for the characterization of functional genes regulation and the interpretation of the result of genome-wide association studies. Toyin, the floor is yours. You're welcome, and then you can take it up from there.
All right, thank you very much. So my name is Toyin Abdulsalam. So I don't need to introduce myself anymore since really, let me make exactly. So my bioinformatics journey actually began at IITA, where I worked as a bioinformatician for some years. And this was where I started to learn bioinformatics for the first time with Dr. Andreas Giselle. And from this point, I began to delve into other fields. I currently work with Data Lead Africa, where we give bioinformatics support for scientists and graduate researchers. And I also currently work as an e-research analyst at Intersect in Australia. Then I'm currently also studying for my PhD. I'm in my third year. And I also work with this lab, this Center for Elderly Brain Aging. So I would like to just talk about my current research that I'm currently doing. As we know that the world population is aging, and yet we still do not fully understand the underlying mechanism that is involved in human aging. Although we know that many age-related complex traits and disease, as well as longevity, they have very low to moderately heritable traits, and which gives a hint that genetic variation could play an important role in this. GWAS over the years has been incredible in identifying this association of genetic variation with different phenotype traits or disease. However, GWAS does not identify the causative variant itself that is responsible for traits or maybe an increase in a disease risk. What it does is to point to the region or the loci in the genome that harbors this causative variant. So identifying this variant is only the first step in understanding a disease and an eventual development of new treatment, for example. So we need to understand the downstream molecular effect of this identified genetic variant, because even for a variant within a gene, it's difficult to predict what the result will be. So the protein coded for this gene might be damaged or completely abolished, resulting in a loss of function, or it may have acquired a new function. And yet genes only make a small part of our genome. And interestingly, most genetic risk factors associated to complex trait and disease are found to be located within non-coding regions of the genome. And these variants can have a regulatory role that affects, for instance, the level of genes, protein, or DNA methylation. And so because of this, my research, this particular research, is aimed to examine the gap in knowledge regarding the functional role of SNPs, particularly those that are located within the non-coding regions of the genome. And so by elucidating how these genetic variants can potentially contribute to changes in gene expression, protein levels, and DNA methylation level in older adults, we want to use this, we're using a multi-omics approach to actually unravel this mechanism. So through the findings from this current research, medical researchers will be able to study this disease better and create therapeutic solutions that will improve clinical outcomes. And so just to give a background, so generally, I would come back to this diagram much later, but just to tell you that expression QTL is when we have SNPs that affect gene expression, and they are located within the non-coding region based on proximity to this gene. They could either be on the same chromosome or at a completely different chromosome. And regularly, I think standardly, it's usually between 1 megabase upstream or downstream of the transcription start site of the gene that is affecting. So the workflow for this project generally looks like this, and I will break it down as my presentation goes on. And so what we did was to, we have this study called the Sydney Memory and Aging Study, which is a longitudinal study. So we used a subset of this study, of which 445 individuals that had gene expression and genotyping data, where we have the average age of these individuals to be about 83 years. And out of this, we have about 209 males. Also, the subset of this data after quality control had about 18,000 expression probes. And then also, so and then as well as about 700,000 SNPs that was available after quality control. And so we did a genome-wide EQTL analysis too, using a linear mixed model, which was implemented in Gamma, and we adjusted for covariate like sex, batch, age, sex, batch, RNA integrity, and blood cell count. So the result of the genome-wide EQTL analysis, which is for the discovery study, identified a total of 13,000 significant associations, out of which we had 10,000 unique expression QTLs, which corresponds to a total of 1,200 probes and 1,000 genes. And just like I said before, we're defining the cis and the trans as to those that are less than 1 megabase of within the transcription start site of the target gene. And then those that are beyond this window, but are also, or they are also on a different chromosome. These are the two conditions we're targeting with this. So what we did also was to break down how this number of probes are associated to SNPs across the chromosome. And so the discovery results found this EQTL probe pair, SNP probe pair association. And then this gave us just an idea of how these expression QTLs were around the genome. So what we did was to find this list, this expression QTL, and then we wanted to know where they are located within the genome. So we saw that about 52% of these cis-actin EQTL were found within the introns, or they were downstream of a given gene, which were about 13% of these. And of those that fell within the coding regions of the gene, you have about 166. Majority resulted in synonymous variant, which were about 50%, and those that were missense variant were about 47%. Also, for the trans, likewise, we tried to find the location of these expression QTLs within the genome, and we found that also 38% of most of these EQTL were found in introns, and about 21% were downstream or upstream of the gene variant. Also, a small percentage also fell within the coding region. So because also we checked, because transcription factor binding site can change, SNPs can change the transcription factor binding sites. And so we examine if these identified EQTLs can affect the transcription factor binding site. So we used a tool called SNP to transcription factor binding site, and we discovered about out of the 10,000 unique cis-EQTLs, we found a total of 1,750, which were cis-actin, and then we found about 118 that were trans-actin. So all of these were creating a new transcription factor binding site, thereby changing the position weight matrix score, which means that it's creating a new binding site and in such a way can affect the binding of the protein within that region. And so if you look at the plot on the right, it shows for each of the number of variants we had, the binding sites. So this is for the trans-actin EQTLs. So we found about 4 transcription factor binding site itself. I mean, yeah. And also this, the binding site we found more in cis-actin expression QTL than the trans-actin expression QTL. So we further, just to check and see, so we did a pathway analysis of the E genes. So the E genes are the genes that are associated with the SNP, because so the probes or the genes. And so this revealed one major significant pathways, which is the lysosome pathway. And lysosome pathway it plays a pivotal role in a plethora of cellular processes, which includes response to nutrient availability and composition, stress resistance, programmed cell death, plasma membrane, and so on. And also, it's known to be associated with many age-related pathologies like Parkinson and Alzheimer's disease, as well as a decline in lifestyle. And so for us to look further, we also checked again if these were enriched in previous GWAS studies, and we found about 112 of these expression QTL have been previously reported before to be associated with age-related disease traits, like all of these that we found. And so so this work is still a work in progress. We're just about halfway done, and the results for this study should be published in about a month's time. And then the other part of the work is so what we did again is to replicate the discovery the discovery results in other independent cohorts, like the old Australian twin studies. And this study had about 218 participants with gene expression microarray, particularly, and with genotyping data. And so what we did is to overlap SNP probes of all significant SNP probe combination for both cohorts, but I mean the Sydney memory and the old Australian twin study. Also, we replicated with in this consortium of architecture of gene expression, and this database, this study had about 5 different cohorts with more than 2,000 participants having gene expression and genotyping data, as well as the Westra study with 4 different cohorts, which had more than 5,000 participants with gene expression and microarray data. So all of these studies use the same genotyping method. That's why we're able to replicate them. So for between mass and old, we replicated about 7,000 expression QTLs, SNP probe combination available. This number was common between both cohorts, and only about 7,000 out of these, which amount to about 96%, was replicated, which is quite high. 96% of cis and 4% of trans. Only about 257 was not replicated. Also, between the Sydney memory cohort and the consortium of the CAGE consortium as well, we had about 8,000 SNP probe pair that matches, that overlaps between both cohorts. And we did a replication analysis which produced about 8,000, amounting to about 98% of replication of trans acting EQTL and 2% of cis acting EQTL and 2% of trans acting EQTL. And we had about 313 that was not replicated. So we use this as our significant level to define whether this is significantly replicated or not. So we use the available data. We just divided by 0.05 divided by the available data and so on. Also for CAGE, the same thing. However, when we overlapped our results with the Westra data, which had a very, very large sample size, we found that just less than 1% of the unique EQTL from the Sydney memory, which is the discovery EQTL study, was overlapping with the Westra study. And this would be because the data comparison is very small. And so, however, just to see that this is consistent with what we got from the other 2 replication studies, you see that the numbers, they don't really differ much. It's still within the same number as well. We found about 8,000 cis EQTL and about 64 trans acting EQTL. So we looked for all of those SNPs that were replicating across all the cohorts at the same time. We found about 5,000 expression QTL that are replicating across all these cohorts. And so what we intend to do is to look at all of these expression QTL that are replicating across all the cohorts. So currently, I'm trying to identify protein QTL in a range of apolipoprotein levels, particularly A1, A2, B, C3, H, using GWAS from mass, OATs, and other source that measures, that use the same method as the mass and OAT. And one particular one which we already submitted a data request was this Hunter community studies, very similar. So next is to identify all of these protein QTLs, and then we'll also want to look at a methylation QTL for this same apolipoprotein. Of course, I know that there is a limitation with this, and the reason is because we are only looking at apolipoprotein, right? Because we're not looking at protein for the entire studies and for the entire cohort, because the sample actually does not exist. However, we plan to get data from UK Biobank to integrate this with the data and try to explain, to see how much of the SNP, which is from the expression QTL, and the protein and DNA methylation explain actually variation in these protein levels. And this is this work should be done by mid next year. And we hope to also publish the second paper also before the end of next year. And so just to conclude, my presentation is that so far we've identified and replicated a catalog of blood expression QTL in older adults. And this will be useful in determining the functional contribution of SNP to age-related traits. Also, the current work will be extended to include other blood eQTLs, such as DNA methylation, blood protein, and even lipidomics and metabolomics. So this will help to unravel further the functional implication of these identified SNPs for aging-related complex traits. Apart from my studies, I'm also involved in research support in Intersect, where I'm involved in teaching bioinformatics and data analysis and computer programming to more than 15 different universities in Australia. And so far, I think I've taught about more than 19 courses, both in Python and R and in basic bioinformatics, as well as Unix shell scripting on high performance computing cluster. And I believe that for me, I'm a product of, how would I say this? I'm a product of multidisciplinary kind of research, which I try to push, because I believe that biological research in any way would thrive when we do multidisciplinary kind of research. Yeah, this is where I would like to stop in case you have questions. I know I was a bit fast.
No, that's okay. No, the speed was okay. Thank you so much for that presentation. And then we will now open the floor for questions. I don't know, can I go first?
Yeah.
Okay. Okay. Sorry. Tony.
Yeah.
In your research design, you are using microarray, and then now you know that RNA sequencing is taking over microarray due to some flaws. What influences you? Why did you decide to go for microarray? Because some low abundance transcript will not be picked with microarray. So you could be missing up some very important transcript or whatever by using microarray. So do you have any reason why you went for microarray? That's my first question. My second question is you talk about your SNP in the upstream section in the non-coding region, right? And then were you able to find out if those SNP were actually affecting the transcription of those genes, or were you able to identify the genes that were actually regulating? Are they acting as an enhancer or they acting as a suppressor? So I think I just want some clarification on those two things. Yeah.
About the microarray, first, I'm not sure because I was not involved in the work from the beginning. I just came in. I have tons of data waiting for me when I came here. But I think one of the reasons why they use microarray would be the fact that they were not, they use these array chips that are very specific in genotyping, and also, for capturing expression that of a particular blood tissue, which they use. However, we also have whole genome sequencing data, and we also have RNA-seq data. But for this present work, and the reason is just because all the data we have, we want to integrate. It's very difficult to integrate microarray and RNA-seq data. So we are doing the RNA-seq analysis as a separate project, which I think I've finished analyzing the data. But because we have more microarray data, which has not been, the data is huge. Nothing is.
Yeah, I guess maybe the reason why is because it could be because maybe the project has been existing for a long time. So so where they were collecting the data, RNA-seq were not actually well pronounced then, well used. So they were using this technology that was available at that time. At the particular, I guess that might be the reason why they went for, because I guess if you're doing research in Australia now, you want to do gene expression, I think most of they would rather prefer to go for RNA-seq. Thank you for that. The second question.
Yeah, so the second question is about the SNPs, right? So so
the
because
non-coding region.
So remember that we were mapping these SNPs also to transcription factor binding site, and then we checked for those that were actually changing the position weight matrix score. They were also located within, some were located within the enhancer region, mostly were located within the enhancer region. And so these are part of those that were actually, replicating across all of these cohorts. So we have this list of SNPs, which we would proceed with the further downstream analysis, which I'm currently doing actually. Yeah.
All right. Thank you for that. Thanks. Dr. Norbert Nwanko, his hand is raised. So can you unmute and then ask a question? The host, can you unmute him? Please, if you have any question, you could just drop your question in the chat.
Yeah, I think it's better they drop at the chat. Yeah.
Okay, but that might take some time, and that I saw it, that could actually prevent people asking questions when you ask them.
The host seems not to be consistently here to be.
Okay.
Yeah, I don't know. Yeah.
That's fine. Okay, then Dr. Nwanko or Norbert Nwanko, can you just type your questions in the chat if you don't mind? Since they said the host is not available to unmute you. Okay, so a question came in. It said, Toyin, can you just share some of your thoughts on the interdisciplinary approach to bioinformatics?
Yeah, like here, for example, you see that I don't have a background in medicine, but I'm currently in Faculty of Medicine. My background is agricultural engineering and computer science. But now I have to learn all of this, so many things. There are a lot of students who are doing a PhD whose background is not in any way related to bioinformatics, nor medicine at all. In fact, the head of bioinformatics here, his first degree was history. And so my point is just that the ability to combine your skills in one field with another, it would help you understand how to solve specific research questions in biology. And so if, for example, you have a biology background, you can just take a time and dive into computer programming. If you do data analysis, it would help you understand exactly what you're doing. And so this idea of having you first degree molecular biology, second degree molecular biology, PhD molecular biology.
Yeah, I think that's one of the major, yeah, exactly. That's one of the major.
It doesn't help our research at all.
Yeah.
because it just makes you
narrow. Yeah.
You're just a professional in one field.
Exactly.
Yeah.
Because at it these days, researchers are now coming more multidisciplinary. Nobody, but in Nigeria, I've seen a situation, someone who is actually a biochemist.
Okay, I think I'm on now.
Okay. Also, let me just quickly, sorry, let me just clear. Sorry, Jordan, 1 minute. Somebody who is a biochemist then got admission for genetic engineering and molecular biology, but the institution turned the person down because the course is not related to biochemistry. So they said you should go for MSc biochemistry. So the way we do research, or the way we do our things in Nigeria now, I think is archaic. I think we should start looking for a way that will embrace what we call multidisciplinary approach to science. All right, sorry, Dr. Rob, no, you can take it up from there.
Yeah, thank you so much. I'm really impressed with his work, and there was a particular area he mentioned. He mentioned the functional implication, including associating his research with age-related diseases like Alzheimer, and I would be glad to find out if he has been able to obtain bioinformatic tool, because this is actually a bioinformatic issue, bioinformatic tools that will help him kind of analyze his data to relate these particular isolations with these functional implications. I don't know whether he has been able to identify the bioinformatic tools that he will use.
Yeah, so the first part of the work is already done. Most of my work, I write my my code, not fully. For example, for the expression EQTL, I was using Gemma, which is a command line application. But however, I have to embed this Gemma into Unix shell scripting because I have, because, you know, it's like doing, I have 18,000 probes, for example. So it's like running, so it's like doing 18,000 GWAS. Technically, that's what it means. And so I have to be able to automate this analysis. So yes, there are available tools to do this, even for the functional classification of the expression QTLs. Yes, there are several tools available. One just needs to decide on what to use. But for me, I'm building along a pipeline for my work, because at the end of this research, at the end of my PhD, the aim is to develop a tool that can help predict age-related phenotypes that can be deployed in clinics. For example, something similar to like genetic profiling, when you give birth to a child, and then you can tell if this child may come up with a disease based on already known SNPs that has been found in that child, for example, that is inherited. And so my main aim is to look at first the genetic contribution to aging, like longevity. So part of my work would be involving the results, my result from this study into, we have a centenarian study, people who lived above, I think the class from 95 years and above, and they have died. Some are still alive, they were involved in this study. So we want to see, there are known longevity genes. And so this is also another area which I'm trying to push my research into. But yes, there are tools, and I've used quite a number of tools, and some I have developed in-house scripts that I use in analyzing my data. Yes.
Yeah, a question here for you. Have you considered obtaining sample for tissue other than blood? What would that be if you were to do that? Plus, is the data available?
Yes, actually, I didn't include it in my slide. We were also checking for this expression QTL for tissue, whether they are specific for tissue, for a particular tissue. So we found out that most of this expression QTL were also found in brain. But yeah, I just did not include this in this research. Yes, we did this. We have data for not just for blood alone. There are postmortem tissues were also taken from the brain. We also have imaging data, which will also be part of, is not part of my work, but I would relate with this. Another group is working on the image from brain from these people at a certain age, because one major problem, for example, in Australia is dementia. Because of the high life expectancy, there are so many people with this kind of disease. And so they would like to be able to make early prediction of this disease and delay the onset of the disease because there's no cure. So such that if it's detected early enough, we can advise lifestyle changes like less of alcohol, doing things that helps the brain, more exercise, fruit and vegetable, and things like this that have been known in the past to help prevent dementia. But there are other cases that we don't have control over, like somebody losing a loved one and became depressed, and all of these things cannot be factored in. Yeah.
Okay, do we have any more questions? I think from the chat, we have no questions anymore, please. So in the absence of any other questions, I think we would like to appreciate you, Tony, for your presentation and for your work. Thank you so much for your time. Our next presenter is Dr.
No.
Is he here?
There's I think he had some misunderstanding with the timing, and probably he will come in tomorrow. So.
Oh, okay. So now, so Tony is the last presenter.
He's the last presenter. Then I take a few announcements, and they will go.
I think I will hand it over to you from here.
Yeah,
so,
thank you so much,
Moses,
Dr. Akinola, for paying the price again to be awake and to ensure that you moderate this technical section for us. We do appreciate this. Thank you for all the support. And of course, I know that as our capacity grows in the near future, we should have some scientists locally that should be able to chair some technical sections and allows you guys in diaspora to sleep well. Yeah.
Right. Thank you so much for the opportunity as well.
Okay. So I want to appreciate the presenters for today. Dr. Samuel and Peter, who is very passionate about genomic surveillance in Africa, and our own Charles Adetunji and Tony also, for your paying the price of staying awake also to make your own presentation. Thank you so much. Maybe I will inform about tomorrow for all of us that are here. Tomorrow is Saturday, and we are going to wrap up tomorrow. And just as we promised that we are going to end on or before 1 today, same will also be the same tomorrow. And we shall also be having some guests from Spain and also our brother in OAU, even though he has his postdoc in Germany. And the abstract presentations will also happen tomorrow, as Dr. Emmanuel Nnamdi will also show us the new virus he discovers through whole genome sequencing. And Norbert also will also make his own presentation tomorrow. Yeah, very important tomorrow is also a very short seminar on starting your bioinformatics career by Professor I.C. Uzochukwu. And I wish you will be around, particularly the students and also the undergraduate and those who are still not yet decided whether to take this path of bioinformatics. So tomorrow will be an opportunity for you to have this. We shall also have poster presentations for 5 minutes each, 2 slides. Of course, we shall moderate all this from here, as most of them have submitted their slides and their poster to us. So tomorrow, within the 3 hours we have tomorrow, we shall really do a lot. And please do well to be part of tomorrow's program as we wrap up. Yeah. So thank you so much. Thank you again for everyone here and the permission to end the meeting today and to say thank you to you for participating. Thank you and God bless. Then those who have not filled the attendance, this attendance is very important. If we are going to give you a participation certificate for this, then you need to come for the 3 days and also take an attendance in the course of the whole thing. So please, if you have not, you can check the chat room. I will not close this room yet, but you are free to go because we have officially ended. And please locate the form and kindly take your attendance before you leave the room, and God bless you. So thank you and remain blessed.